Hello everybody and welcome back to the lecture. So, today we will start a new module, module 6. So, after an extensive amount of biology and biological processes of how proteins are synthesized in biological cells, we are now back again with chemical tools. How chemistry can be used in order to study or in order to know, understand some of the biological molecules, biomolecules. So, in this module we will talk about protein sequencing and solid phase peptide synthesis. So, this module is actually a combination of two topics. One is the protein sequencing and the second one is how can you synthesize short peptide chains or short peptide molecules in your laboratory, and both of these techniques would involve chemical tools. We will use the knowledge of chemistry or the methods of chemical reactions in order to study these two subjects. So, let us start first with the protein sequencing. So, sequence of a protein means which amino acids are connected together in a row, so that you will understand the composition of the protein as well as the specific linear chain of the or the primary structure of the protein. Of course, this is a very important aspect to know, because that is how you can understand or you can kind of predict what kind of function of a certain protein will assume. We have so many enzymes in our biological systems, both eukaryotic cells and prokaryotic cells, and all those biological processes are catalyzed by enzymes, which are basically the proteins. So, it is of course very important to understand or very important to find out the sequence of a protein. So, there are kind of several techniques, 3 to 4 different techniques that can be adapted for protein sequencing. So, before we have seen that how to do the DNA sequencing. We have seen the Sanger's method, we have seen the Maxam Gilbert method that were being developed to know the sequence of a given DNA or a gene. And DNA sequencing was kind of pretty much straightforward. So, in Sanger sequencing or also the Maxam Gilbert method, we have seen that if you have if you just read the gel, if you see the image of the gel, you can pretty much find out or you can pretty much write down the sequence of the DNA or the gene. Protein sequencing methods are not that straightforward, and it involves a combination of methods. So, before going to the actual sequencing of the protein, what most people do is to find out first what is the composition of the protein, what amino acids are involved and in how many quantity. So, at first we need to know the composition of the protein. We have to know the composition of a given protein. Or it can be enzyme, protein or enzyme, it is the same thing basically. So, this will tell you, this means what are the amino acids present in that particular protein sample, and in which quantity, how many of them are there. So, that is called the composition of the protein. Because protein is a very long molecule, so before going to the sequencing, you need to have some ideas about them, that makes the process easier. So, how do you do it? Usually this is done by complete hydrolysis of the protein or complete hydrolysis of the peptide sequence. For example, if we write down a sequence of a protein, alanine, glycine, aspartic acid, phenylalanine, arginine, glycine, just arbitrary sequence. This is a short peptide, actually I have not drawn the whole protein structure or whole protein sequence. So, this is your N-terminal, which means it has a free amine group, N-terminal, and this has a free carboxylic group, which is C-terminal. Of course, they can exist in zwitterionic form. Now, composition of a protein means to know, this is sequence I have written, but if you isolate a sample, you do not know what is the sequence. So, composition of a protein means just to find out which amino acids are present. So, to do that, what is done is you treat the protein with a 6 normal hydrochloric acid, 6 normal HCl, and heat at around 110 degrees Celsius. It is a pretty rigorous condition actually. So, what does it do? It breaks all the peptide bonds, it breaks all the amide bonds present in the protein. So, if I just write the alanine, I am not drawing the stereochemistry. If this is your N-terminal, this would be CO, and this is your glycine with H here, NH, CO, and this would be your aspartic acid, and it will go on. I have missed something, right? CO here, I am sorry, CO, NH, this is your glycine, CO, NH, and so on. So, if you treat this with 6 normal hydrochloric acid at high heat, then it will cleave all the peptide bonds, and what you will get is individual amino acids, this plus a glycine, plus all the others. So, after treatment with 6 normal hydrochloric acid at 110 degrees Celsius, what you will get is individual amino acids. It will break all the peptide bonds. So, that is what is done to understand the composition. So, if you treat this with this, then all the individual amino acids will be formed. So, this is known as complete hydrolysis of the peptide or protein. So, once you have all the amino acids, then what do you do is you try to separate them individually. So, next step is the separation of the generated amino acids. So, in this sequence what would you get? What you are expected to get? You should get alanine in 1 mole quantity or 1 equivalent quantity. You should get glycine individually, 2 of them. If you look at the sequence here, you have 1 glycine here, you have 1 glycine there. So, glycine should be of 2 equivalent, and then you have aspartic acid 1, phenylalanine 1 and arginine 1. Aspartic acid 1, phenylalanine 1 and arginine, that is also 1 equivalent. So, this should be the composition of the amino acids present in the mixture, but you still do not know their identity because they are still in the mixture. Now, what you do, you try to separate these individual amino acids using chromatographic technique. So, usually a chromatography means its chromatographic technique is a separation technique, where you can separate individual molecules. If you have a mixture of number of molecules, then you can separate the molecules into the individual ones using the chromatographic technique. Now, there are a variety of chromatographic techniques, separation techniques available. So, I will talk about those in detail when we will be talking about the modern techniques in that module. So, roughly there are many chromatographic techniques based on the separations can be based on the physical properties of the molecules that are present in your mixture. So, those properties can be based on the polarity of the molecules. So, if you have a mixture of a non-polar compound and a polar compound, then you can separate them using a chromatography based on their polarity difference. Similarly, you can separate the molecules based on their charge or charge density, as we have seen in the electrophoretic diagram, that DNA, shorter DNA with less negative charge, longer DNA with more negative charge can be separated using the gel electrophoresis. So, in chromatography also those kinds of techniques are there, where you can separate the molecules based on their charges. So, here in this case, and all of course, on their pH, based on their pH also, because some molecules will be eluted out at certain pH level. That is another way of separating the molecules using the chromatography. We can also separate molecules based on their size, which is known as size exclusion chromatography. If you have a small molecule and if you have a very large molecule, obviously their sizes are very different, and you can separate these two molecules based on their size difference using a certain chromatographic technique. So, in this case what we use is ion exchange chromatography. Ion exchange chromatography is a separation technique which separates molecules based on their charge and the pH. So, that is why it is called the ion exchange. So, the ions basically. So, if you do this, then you can have versus the time, then you can have the individual amino acids appearing at different time interval. So, for example, I am not going details into the methods how the chromatography is done. So, it can be based on the change of ionic value you can do based on the pH difference. So, ideally what you can remember is that the time. So, if you are running your mixture of compounds, in a chromatography column. Here is your mixture of the amino acids, individual amino A's, and then if your column is packed with the ion exchange material, then based on their ions or ionic charge or pH, these molecules will come out at the end of the column at different time. So, if you draw a time versus the appearing profile, it will look something like this. Here you will see one peak, so this would be might be for aspartic acid, this would be for glycine, this would be for alanine. Glycine and alanine are not very different, only on methyl group. So, they will be coming somewhat close. Phenylalanine, and here little bit further would be your arginine. So, at different time they will elute out. So, obviously, there is a gap in between. So, you can collect them, that will give you the separated amino acids in individual purity. So, that is one aspect. Second is, fine, you have got your amino acids. Now, how to see them? All these amino acids do not have any color, and they do not, most of the amino acids do not absorb UV radiation also, UV light, except the tryptophan and some of the others. So, most of these amino acids are not sensitive to UV radiation. So, you cannot see them. So, what you do is, you actually do a reaction here. Once you have all the amino acids after the hydrolysis, you treat the mixture of amino acids with ninhydrin. So, do you remember the structure of ninhydrin? And we have used ninhydrin to find out the amino acids. If you have amino acids, how to determine or how to characterize the presence of an amino acid, that was done by ninhydrin test, right. So, the same thing is done here. I will draw again the structure of ninhydrin. OH. So, this is your ninhydrin. Ninhydrin, if you treat with free amine group, which is present in the amino acids, I am writing R in general, and then you have the amino acid. Then if you go back and look at the mechanism of these reactions, what is the product finally you get is this. Six membered. I guess it was something like this. So, this one was your product, and that has intense color. It was intense purple color, right. Except proline, which proline gives yellow color, orange to yellow color. So, basically you get colored compounds. When you treat the amino acids with ninhydrin, you get the colored compounds. So, what is done is, once you have this profile and separate them, each of them, I will write the profile again. So, you have separated aspartic acid. Now, you have all the separations there. You have glycine, you have phenylalanine, you have arginine and all this. So, each of them are now treated with ninhydrin and you get a color. So, now by measuring this color of course is that will absorb UV radiation, UV light. So, you can see the UV absorbance and can quantify it. So, the y axis I was writing here, the intensity of this peak would now be visible when you have, if you now measure y axis the UV absorbance ABS, I am writing for absorbance, then you can see the peaks properly and their intensity, how much the intensity, what is the absorbance, how much is the absorbance of the individual amino acids. Aspartic acid would be here and then you can see the glycine, then you can see the alanine, you can see the phenylalanine and you can see the arginine, SP, glycine, alanine, phenylalanine and arginine. So, this is based on your time or pH variations. So, now by measuring the intensity of the absorbance, ideally it is the whole peak area calculation, area of the peak. If you calculate, then from there you can find out which amino acid is present in how much quantity. And from this spectra you can find out the glycine will have the 2 equivalent quantity or the intense peak area would be double compared to all the other ones, because glycines are present in the whole peptide that I have written. So, that is how you can actually find out about the composition of the amino acids present in the peptide or in the protein, and you can also know the quantity of the individual amino acids. That is the first step. Now, once you know that, after that you go for sequencing method. So, now starts the real protein sequencing. So, as I have mentioned, sequencing is about which amino acids are connected to each other or the primary structure of the protein. Now, protein is really big molecule. A large molecule contains 200, 300 amino acids in a protein chain, and the methods that are developed, I will first describe the methods and then actually we will this thing I will talk later, that you have to actually cut the proteins into the shorter peptide versions to use them for the peptide sequencing or protein sequencing. So, there are 3 major methods, all are chemical methods used to understand the sequencing of a protein. The first one is, again Professor Sangars came here for our rescue. The first one is called the Sangars method. Sangars protein sequencing. The second one is called Edman degradation. And the third one is not a chemical tool, it is an analytical tool using the mass spectrometry. So, nowadays Sangars degradation method is the most widely used for the protein sequencing, You also have the automated machine, just like the PCR, just like DNA sequence I have talked about. For protein sequencing also, there is automated machine pre-programmed, but based on the chemistry of the Edman degradation. So, this is the most widely used method for protein sequencing. Nowadays, mass spectrometry is also used as the major technique to understand the sequence of a protein. So, this is of course, the using the high end mass spectrometry. Okay. So, let us talk one by one. Sanger's method. The method was developed close to 1950, 1945 I think, long back. In fact, Sanger's protein sequencing method was one of his first work. Much before this was done much before the DNA sequencing was developed. So, it is purely a chemical method. In DNA sequencing, we had seen the Sanger's method was a constructive method. It was not touching your parent sequence of your of the gene. But here the protein sequencing method is a destructive method. You do not get back your compound. It is a destructive method. So, you cannot rerun the system. Primarily, Sanger's method is developed to know the N-terminal sequence of a protein. So, if you have a protein which has the N-terminal, this is the C-terminal. So, Sanger's method primarily tells you determines the N-terminal amino acid. In a protein. And this is the first method that was developed, or who has made any attempt, that was the first attempt made towards knowing the sequence of a protein. But it was of course, at the beginning it was only for understanding the N-terminal sequence of the protein. So, as I have said that it is a chemical technique. What he has used is a chemical molecule, this molecule. It's fluoride, it's nitro. This is an aromatic compound. The name is, this is 1, 2, 3, 4. So, it is 2,4 dinitro fluorobenzene. This is the molecule Sanger has used to know the N-terminal amino acid. So, how was it done? I have to draw the structure again. Why he has used this molecule? The molecule was used to label the amino acid. If you have a peptide or a protein, so I am not drawing this is your protein or peptide, which have the N-terminal amino acid will have the free amine group. So, what this will do is, this is a nucleophile. So, it is basically a nucleophilic substitution reaction to the aromatic ring. Now, obviously, you know that the nucleophilic substitution to aromatic compounds is not very facile. is not at all facile actually. The reason is, if you have a nucleophile, which means that it has excess electrons, that would be attacking. Now, your aromatic ring also is rich in electron. So, there are 6 electrons, 6 pi electrons in the aromatic ring. So, it is already rich in electron. Therefore, reaction with the another nucleophilic center, another electron rich center will not be facile. There is no reactivity here. What you need to create is an electron deficient center. So, usually in the aromatic ring, nucleophilic substitution is not facile. If you have just benzene, if you try to react with a nucleophile with a distinct minus charge, this reaction will not occur, because this is electron rich, this is also electron rich. But here this molecule undergoes quite rapid nucleophilic substitution. The reason is, since you have the nitro group, nitro groups are highly electron withdrawing groups. So, they will pull the electrons towards themselves, that would create the electron deficiency in the aromatic ring, and therefore, you have also the fluoride, which is also have minus I effect, that pulls the electron density towards itself. So, this carbon here becomes electron deficient, or overall the aromatic ring is also electron deficient. That is why this nucleophilic substitution can occur. So, it is basically the amine would react, amine would attack this carbon and fluoride would be eliminated. So, nitro, here is your amine and the rest of the peptide. So, for this given compound, due to presence of electron withdrawing nitro groups, the nucleophilic attack of the NH2 group of amino acid, which is obviously the N-terminal amino acid, becomes facile. That is why this reaction occurs. Now, once you do that, once you get your compound, now how to know the identity of the N-terminal? You again go back to the previous method, 6 normal HCl, complete hydrolysis at 110 degree Celsius. So, if you treat this molecule with 6 normal HCl under heating condition, then it will hydrolyze all the peptide bonds. So, that will hydrolyzes all the peptide bonds, and you will be left with, I should draw this properly. This amino acid plus all the other amino acids AA. All the other amino acids present in this peptide would be in their free form after the hydrolysis. The first one, the N-terminal one, would be attached to this 2,4-dinitro aromatic compound, and this is colored. So, this can be separated out, eluted out. So, this will have a distinct appearance compared to all the other present in the mixture, and of course, it can be easily separated also. So, once you separate this, you can know from your standard curve, if you have a standard, that which amino acid will appear at which position. If you have done individually with all the 20 amino acids reacted separately with this, then you will know exactly where in the elution that particular amino acid will appear. So, from that comparison you can find out the nature of the R, or you can find out what is the amino acid present in this molecule. So, that is how you can determine the presence of the N-terminal amino acid. For all the other amino acid, you cannot at this moment. You can know the composition of course, but you cannot know the sequence. Since the NH2 was free at the N-terminal end, you can be certain that this molecule is your N-terminal amino acid. So, that is how Sanger's method was used to find out about the N-terminal amino acid of a given protein. Now, the question is it important? In that time actually it was very important, and it was, as I have said, it was one of the first attempt ever made to find out a sequence of a protein using this technique. So, if you remember, Sanger has solved the structure of insulin and found out the complete structure of insulin. This is one of the technique that he has used, one of the prominent method that was used to understand the insulin structure. So, if you have insulin, so what we know now is Sanger's method can be used. Sanger's method can be used to estimate the N-terminal amino acid. Using this method, Sanger found out that insulin has 2 N-terminal amino acids. So, if you write a peptide sequence, usually if it is a single chain, that will have only one N-terminal amino acid which has a free amine group, and that will have only a single carboxylic terminal end. But in insulin, what Sanger has found out, that instead of getting one amino acid here attached with the aromatic ring, there are 2 compounds that were attached with the aromatic ring, and upon characterization he came to know that these 2 amino acids are, so these 2 N-terminal amino acids, those were glycine and phenylalanine. Phenylalanine, pH, glycine, and this is PHE. 2 compounds were there, which obviously means that the insulin has not a single chain of protein, it should have at least 2 protein chains combined together to form the whole protein. Not only that, Sanger has also found out that if you estimate their quantity, glycine was in 2 equivalent in number, phenylalanine was also 2 equivalent in number, 2 molecules per unit per unit. That means, insulin has 4 N-terminal amino acids. 2 of these N-terminal are glycine, 2 of the N-terminals are phenylalanine, which essentially means that insulin has 4 chains of peptides, 4 long peptide chain or 4 protein chains present in the whole protein structure, in the whole protein structure. So, it has to be a combination of 4 subunits of protein combined together, that is insulin, and that was really a big discovery, big development in that time in understanding the structure of the insulin. Just using this method, using this chemical compound which is 2,4-dinitrofluorobenzene. So, nowadays actually, so as I have said that fluorobenzene is, it absorbs UV light. So, you can see it in the UV chamber, or you can estimate its quantity if you do the UV spectroscopy. So, nowadays we do not use the fluorobenzene, that is been replaced by fluorescence molecule. So, instead of using that 2,4-dinitrofluorobenzene, now the fluorescence molecule are being used to tag the N-terminal amino acid. One of them, there are 2 of them which are popular. CH3, CH3, this compound is known as dabsyl chloride. This shows fluorescence, or there is another one called dansyl. What is the structure of the dansyl chloride? Yeah. Similar functionality. Here is the SO2Cl, in this case here is the SO2Cl, and this is the secondary amine. So, this is known as dansyl chloride. Both dabsyl chloride and dansyl chloride show fluorescence, and both of them react with the free amine present at the N-terminal amino acid. Simple reaction, nucleophilic substitution, chloride out. I am just writing this. CH3, CH3 in double bond in, NH. Now, you can draw the other peptide. So, R1, CO, NH and so on, followed by hydrolysis. That will cleave your peptide bond here, and you will be left with CH3CH3, NH, R1, COOH, the first amino acid, the N-terminal amino acid, and then all the individual amino acids will be there. So, your first N-terminal amino acid would be attached to the fluorophore unit, and you can see just by reading the fluorescence you can identify the structure of the compound, or you can also find out what is the amino acid present at the N-terminal. So, that is about the Sanger's method, which is used for the determination of the N-terminal amino acid, but it does not tell you about what are the sequence of the other part of the peptide or other part of the protein. For that comes the Edman degradation. Edman degradation. Edman degradation is the most widely used protein sequencing method that tells you about the complete sequence of the protein. Most often the protein sequencing is done in combination of Sanger's method plus the Edman degradation method. Sanger method is used to find out the N-terminal amino acid. Once this is done, then the Edman degradation will come into the picture. So, this is also a chemical method. And a destructive one. Which means your compound or your protein sample is destroyed, you do not get it back. How is it done? It also uses a specific chemical reagent, which is this aromatic this is phenyl isothiocyanate. This is the molecule or the reagent that is used to react with the protein. So, let me show you the reaction first, and then we will go for the sequence wise how is it happening. So, if you have, let us take a peptide sequence CO, this is NH, this is R1, this is R2, R1 I have taken as alanine, this can be R2, okay, let us say this is a glycine with H, CO, NH, and then CO the rest of the peptide. Why I am writing a little bit sequence you will know shortly, because the reaction happens here. So, first thing first is it is a nucleophilic attack of the NH2 N-terminal amino acid here, that will ultimately comes here and then it comes back, whatever you get a N minus, which will abstract the proton from here. So, it is a minus H plus and plus H plus. So, this becomes NH, this carbon double bond S, this carbon is connected covalently to this nitrogen. So, it would be NH here. I am not drawing the stereochemistry of the amino acids. So, they are all L configuration, and then it will be here NH, this is your glycine CO NH, CO NH, this should be the, I am sorry, glycine, this should be something else, let us say phenylalanine. and then the rest of the peptide. CO, I think we can go here peptide, do not need more. So, this is formed. Now, once this compound is formed, there is another internal reaction that occurs. Here is your NH. This lone pair or this bond breaks down, it will form a double bond that makes this sulfur electronegative, and this reacts with this carbonyl compound. It reacts here and it ultimately breaks this bond. So, this reaction or this cyclization would break the first peptide bond that is present here. This bond would be chopped off, and if I can write here, it will be this will be N, here is the double bond, this should form a 5-membered ring, this should be your S reacting with CO, O, this is chopped out. So, now is CH3, now comes the NH here, yeah, this is N, this is H. This would be formed, plus it is cleaving here, the rest of the peptide is, it will make another this NH2 free, NH, you had glycine, H, CO and the peptide. So, this is the essential part of it. It will because of this internal cyclization, it cleaves the first peptide bond, that results in formation or exposure of a new amine free group, which is attached to the rest of the peptide, plus this molecule with the first N-terminal amino acid, which is alanine in this case. And this is a small molecule which can be, this actually this compound further gets rearranged into a stable product. Write this down, N, S, H, this is CH3 and double bond O here. What happens is this double bond through the nitrogen that reacts here, and this sulfur double bond, this bond gets back. So, you form the carbon sulfur double bond, you attack this, that gives you this compound. So, as you get this stable ring cyclized product plus the peptide attached with the other group except the first one. Now, this is eluted out because it is a small molecule. This version is a long molecule still present and it can easily be separated. So, this will be eluted out, and from its characterization you can know what is the first amino acid that was present. And now, you are left with the other part of the peptide with an exposure of another amine. Now, once this is eluted out, this one will start reacting with the Edman reagent again, and it will continue. So, one by one you will get a cleavage. Now, you are getting the sequence. So, it is happening one by one, not all at the same time. So, first this is out, second would be glycine would be out, third the third amino acid would be out, something like that, it will go on. So, I will quickly show you the sequence, the scheme, how this is happening. Let us say this is the first amino acid, this is the second amino acid, this is the third, this is the fourth, fifth. This is the first one which has a amine free, this last one. Now, you react with the pH, N, double bond C, double bond S, phenyl isothiocyanate, which I am representing by this symbol. In book also this symbol is used. So, whatever I am teaching for this protein sequencing, you can find it out in the Stryer book, biochemistry, written by Stryer. So, this then what will happen? 2, 3, 4, 5, and here you will have this conjugated to the amine group, followed by, so this step is known as labeling, because you are labeling your protein with the phenyl isothiocyanate, a marker group. So, once the labeling is done, the second step is called the release, because this is where you are breaking it. It will break this bond, this is formed, you can see here, the first one is formed and the rest is there, and this is called release because you are breaking this bond, keeping the rest of the peptide, releasing out the first amino acid, plus you get the amine free of the second one, 3, 4, 5. This is eluted out because this is a small molecule compared to the large protein. This is a very small molecule, so it is easy to take it out, and you are left up with this. Now, again this is exposed. So, in the next scheme I will start here in H2, this is 2, 3, 4 and 5. Again you react with this, it is all happening in the same mixture, you do not have to do it separately. Once the other part is out, the smaller part is out, the rest of the thing you already have phenyl isothiocyanate in your solution. So, it will go on. So, it will be 2 that is connected, that is label, 3, 4, 5. So, this is labeling step again, followed by release, that will release number 2, plus you will have free amine of the 3. This would be eluted out again and characterized, and then it will continue. Right. So, all these things are done online. I will write this term, online separation. The moment the small molecule is formed, it will be released or it will be eluted out, and the rest will go for the next step of the reactions. So, you are at the same time you are characterizing the small molecule, what is the identity of this amino acid, plus the other part of the reaction is going on at the same time. So, that is how sequentially you are breaking or you are sequentially cleaving individual peptide bonds one by one, and you can understand their nature by the online separation and the characterizations. So, that is about the advanced degradation, which will tell you the full sequence of a protein. Thank you.